Our next talk is from Sydney, and I hope I pronounced your last name right, Prangi? Prang. Okay, Sydney Prang from UC Irvine. Take it away, Sydney.
Gotta lower it because I'm really short. Okay. Hi, everyone. My name is Sydney Prang. I'm currently an incoming fifth-year PhD candidate in the Thompson Peer Lab at the University of California, Irvine, and I've been a CERM scholar for the past 3 years. So I'd just like to thank CERM, first of all, for funding me for these 3 years, because it's been really integral to being able to work on this project, in which I'm investigating dendrite regeneration in the context of neurodegenerative conditions. So for the non-neuroscientists in the audience, this is just a general schematic of a neuron, and neurons are the fundamental unit of the nervous system, and they play this really important role of information signaling throughout the body. And to do this, they use two sorts of neurites. One of these is dendrites, which are used to receive signals, and then axons, which are used to send signals. And so both axons and dendrites are really important for the important function of signaling throughout the body. However, across many different neurodegenerative conditions, you get loss of dendrite architecture and simplification of dendrite arbors. So what I'm showing you here are models of 4 different neurodegenerative conditions. The top left is a Huntington's disease model, the top right is an ALS model, the bottom left is a Parkinson's disease model, and the bottom right is a spinocerebellar ataxia type 1 model. And wild type neurons are on the right, whereas the disease condition neurons are on the left, whereas the disease condition neurons are on the right. And hopefully you can appreciate that the dendrites of these neurons are quite simplified. And importantly, these dendrite defects and this dendrite simplification and degeneration happens early on in disease stages before sort of the mass death of neurons that are generally associated with the late stages of neurodegenerative disease. So because dendrites are critical for this function of neurons to be able to signal, likely these defects are related to and critically contribute to the pathogenesis of these diseases. And so our lab actually studies dendrite regeneration, and we use this really fun little organism called a Drosophila melanogaster, which is a fruit fly. And in these animals, so what I'm showing you here is a Drosophila larva. If you look at their peripheral nervous system neurons, they're actually able to regenerate their dendrites if you completely remove them. So in the bottom left corner, you'll see a neuron where all of the dendrites were removed, and then by 72 hours after that injury, the neurons are able to completely regenerate a really beautiful dendrite arbor. And similarly, it's been shown in zebrafish models. If you look at the zebrafish spinal cord and you also do acute dendrite injuries, those neurons are also able to regenerate their dendrites. And so we wanted to know, can we take advantage of these regenerative mechanisms to regenerate the dendrites that are lost in neurodegenerative disease? And in order to answer this question, like I said, we use the Drosophila melanogaster larva as our model system, and specifically we're using these sensory neurons in the peripheral nervous system, which are called dendritic arborization neurons. And the neurons that I'm looking at are those ones that are in green. They're the class 4 neurons, and they have these really beautiful big dendrite arbors that are really complex, and they sort of tile on the larval body wall, so they're a really great system to be able to actually injure neurons and see what happens following that injury. And the way that we're able to do injuries in the system is we actually take live animals and we use a 2-photon laser to really specifically injure individual dendrites of these neurons. We could also injure the neuron cell body itself or the axon. It's just a really great system to be able to study these processes. And so in order to study how regeneration in the context of neurodegenerative disease, we needed to pick a sort of a neurodegenerative disease that we could model easily in flies. So we decided to look at polyglutamine diseases, which are a family of inherited neurodegenerative disorders that are caused by an expanded CAG repeat in a specific gene. And these are disorders that have been extensively modeled in flies, and we've learned a lot about these diseases from looking at them in flies. So they were a great model for us to start with. And just to explain a little bit about the pathology of these diseases, in someone who doesn't have the disease, you'll have a specific gene with a wild type normal number of these CAG repeats, which are also they encode glutamine. So interchangeably, they're called like Q or glutamine repeats. So I might use those terms interchangeably. And then because you have the normal number, you get the correct wild type RNA and protein, healthy neurons, and a healthy nervous system. However, in people that do have the disease or inherit this disease, you get an expansion of that CAG repeat region to a pathogenic number of those repeats. Okay, and you get a pathogenic RNA and protein, which causes a lot of issues for the cells and also can aggregate. And that leads to neurodegeneration and degeneration of those dendrites, like I showed before, and then eventually neuronal cell death and a pathogenic nervous system. So when we look at when we take these human genes that have the expanded repeats and we model them in our neurons, on the top you'll see a wild type neuron, which has this big, nice, complex dendrite arbor. And on the bottom, you'll see what happens when we have an older neuron that has this human protein with the expanded polyglutamine repeat modeling spinocerebellar ataxia type 1. And that's the disease I'm going to focus on today. But we've done all of these studies with various different polyglutamine disorders. So you can come to my poster if you want to see more. So if we take these neurons that are eventually fated to degenerate their dendrites and then eventually die, we wanted to ask, what if we instead intervened and did a single dendrite branch injury at this sort of earlier time point, trying to catch them when they're degenerating and see what happens? Would we be able to protect or regenerate those dendrites that are being lost? And excitingly, we were able to do that. So what I'm showing you here is on the left is our uninjured control neuron, and we just sort of normalize for the fact that an entire dendrite branch is missing in our injured condition. And then on the right is our single branch injured neuron. So that's the neuron where I actually did the 2-photon laser injury, and the red arrow is showing you where I removed the dendrite branch. And hopefully you can appreciate that these neurons look quite different. The single branch injured neuron looks quite a bit more branched, and when we do the quantification, that is indeed what we see. The single branch injured neurons have significantly more dendrite branches than their controls at the same time point. And when we look at individual neurons and what's happening actually between the time of injury and the final time point that we looked at regeneration, we see that the controls are overall having this negative loss of branches, whereas our injured neurons have this stabilization of branches. So instead of having this negative trend, they have a slightly positive trend overall, and it's a stable growth. There's no loss of branches overall. So we were able to sort of rescue that by just doing this injury. So we were able to find that a subtle dendrite injury to a single primary dendrite branch was able to protect against this dendrite degeneration in these neurodegenerative models that we were looking at. And we were able to recapitulate this effect in models of SCA1, SCA2, SCA3, a model of HD, and also a model of tau-induced degeneration. So it seems to be somewhat applicable across neurodegenerative disease types. And so the next thing we wanted to know is, was this actually specific to dendrite injury, or was it just something that would happen as a result of generally activating regeneration in these neurons? So we wanted to see if we did an axon injury, which is another important field that a lot of people study, but in our lab we care about dendrites. So we wanted to see if axon injury could also cause this response. And excitingly for us, because we love dendrite regeneration and think it's important and we should study it, we didn't see anything with the axon injury. So when you look at the uninjured neuron on the left in the purple, and the neuron where we did the axon injury, also called an axotomy, in the yellow, they don't really look that different. And when we quantify this, we also don't see any significant difference in the number of branch tips. When we look at the uninjured neurons, they're overall losing dendrite branches, and similarly, the neurons where we did the axon injury are also losing branches over time. And so injury to the axon does not cause the same neuroprotection, but injury to the single dendrite branch does. And so what I've been working on for a lot of the recent time in my PhD has been looking at what is really the mechanism that's causing this. And the first thing that we wanted to look at was just the cytoskeleton, because the cytoskeleton is really important for neurons to keep this structure and for them to function properly. So we looked at lots of different ways to look at microtubule structure and microtubule dynamics in these neurons, but we didn't find anything interesting. So instead we moved on to looking at actin. And so we looked at this actin reporter and just looked for stabilization of actin in these neurons. And hopefully you can appreciate that the uninjured neuron, so this is once again our model of spinocerebellar ataxia type 1, and the uninjured neuron, you have this low level of this actin reporter in both the cell body on the top and the dendrite branch on the bottom. But when you look at the neuron where we did the single branch injury, you get this strong enrichment of F-actin in the cell body as well as in the dendrite branch. And when we quantify that, we also see that this is a significantly increased enrichment of F-actin in both the branches and in the cell body. So overall, injury to the axon, no neuroprotection, but injury to a single dendrite, you do get this neuroprotection that's underscored by this increase in F-actin in both the cell body of the neurons and the dendrite branches. And if you're interested in learning more about this work, it's currently up as a preprint in bioRxiv, and we're currently going through the resubmission process and responding to reviewer comments, so hopefully it will be out soon in a peer-reviewed journal. And I just want to thank everyone in my lab, especially the undergrads who really have been the workhorses for helping me analyze all this data. All the neurons that you see in are hand traced, so that's hours of work. So I'd like to thank Hugh, Isha, Daria, Grace, Jayla, Lee, and Anjali for all their critical work on this project, as well as Adila Syed at the UCI Optical Biology Core. Without their microscopes, we would not be able to do any of this work. So thank you so much, and I will take questions.
Hello, great talk. My question is, do you know if this regeneration also happens in adult flies, and not just in the larva?
Yeah, so that is a really great question. So adult flies, these same neurons are present in adult flies, not the specific ones that I'm looking at, but another type of class 4 neuron called the V prime ADA neurons. I have tried these experiments in adult flies, but the natural regeneration of these neurons is pretty much limited to like 1 to 3 day old adult flies. So, and if you express these proteins in those flies from the day that they close, you don't get any neuron degeneration until they're like 20 days old. So we've been trying to, for a while, working with how to figure out how to get the degeneration to happen in the young flies so that we could do these experiments, but we've not yet figured out how to get that to work. So it's on my like to-do list after we finish resubmitting the paper.
All right, thank you.
Hi, cool talk. I'm not a neuro person, but I'm curious. You have the single dendrite that you are injuring. Mm-hmm. Have you kind of like gauged what your parameters are? Like, can you injure a couple of dendrites and have the same effect, or could you injure one of the little branches coming off of the dendrite? I don't know what those are, but what's kind of like the parameters for engaging this sort of regeneration?
Yeah, great question. So I have done those experiments. So we also do this like kind of typical, or I guess bread and butter assay in our lab is called balding, which is where you remove all the dendrite branches of these neurons. I have done those experiments in these neurons, but at the later stages when they've already started to sort of lose dendrite branches, they don't regenerate super well. They're able to do a little bit of regeneration, but not anything not a big, nice dendrite arbor. I've also done experiments where we just injure, it's called like a terminal branch. So you have your primary branches and then secondary branches, and then the terminal branches are just the last branch on the tree. I've done experiments where I look at injuring just a terminal branch near the cell body of the neurons, and I don't seem to get the same response. It doesn't induce as much of a regenerative response. So it seems like there needs to be enough of a signal that the neuron is able to respond. But I haven't done anything in between those, I guess, two extremes.
Cool, thanks.
Yeah. Hi, great talk. I was wondering if you've looked at ways to upregulate F-actin either with small molecules or in some other way to rescue the phenotype without inducing injury. This is thinking like longer term for a therapy.
Yeah, so that's definitely, the most important thing, right? If we don't know what the mechanism is or what to target, it's not going to be something that's applicable for any therapeutic targets. So that's actually what I'm working on right now. Once we finish resubmitting the paper, I'm working with looking at a bunch of different actin modulatory proteins and seeing if we, upregulate those or downregulate those, can we either improve or lose the phenotype, and then also you could do it, in the absence of injury and see how that affects the neurons as well. So definitely something on my to-do list.
Great, thank you.